Okay, so good morning. Um, Thanks to all of you who submitted the subject to. Okay, um, I'm hoping I guess I got from maybe 7 students. I'm guessing the other ones also got the results, and I got. I think the latest I got was at 3 a.m. Someone sent me like Joshua. I was like awake and I thought, should I respond to him or not? And if I responded, maybe that would scare him more. So I decided not to. But thank you. I hope you didn't wake up just because of me. All right. So today we'll cover about PCA, ICA, and how we use the analysis. Okay. And so these come under multivariate analysis, right? And in multivariate analysis, there are multiple variables which are often represented by a single vector variable that includes different variables, right? So you could have like x1t, x2t, xmt for I equal to m, right? So for the resting state data or for even task data, what would x represent? So in fMRI, what would x represent? So I told the other dimension is the time. So what would x represent? Anyone? Voxel, right? So it would represent voxels. Um, so there are a number of voxels and they are capturing some common properties, and we have to basically decipher this, right? And T stands for transpose matrix, right? So it could be in row or it could be in column, doesn't matter. So the variable x would be the image, right? Is composed of m variables, each containing n, where like n is the number of observations. In signal processing, the observation are time samples, while in image processing, they are pixels, right? But in fMRI, like x would be voxels, while t would be the number of time points. And so multivariate data, as you know, the Wi-Fi in my hotel is really bad, and even the Digital India is not working. So during lunch, I will share the slides, and if I cannot, maybe I can give the like a USB to one of you, and maybe you can share it. Okay. Right. So multivariate analysis takes into account relationship between multiple variable as well as within the variable, and uses tools that operate on all of the data, right? And the covariance matrix is a multivariate analysis technique, as it includes information about the relationship between variable as well as about the variable themselves, right? So the location of the pixel and how they respond in time is important, but where the pixels are located is also very important. And therefore, a major concern of multivariate analysis is to find transformation of the multivariate data that make the data set smaller or easier to understand. Like, so you have large number of measures, and from those measures, you're trying to find common properties. And the reduced set of variables may even be more meaningful than the original data. The more meaningful variables are hidden or latent in the original data. Right? So this is the mathematical representation where what you have to do is, right, these are, again, like using the same analogy, this would be the output voxel, the time response, this would be the input, right? For task activation, this would be known, okay? What we have to find out is the weight, right? So again, another way would be you have x1, x2 till xm, and then you have a number of measures, right? And the assumption we make is that you can find a set of measures. So let's say you presented two different tasks, right? One, like two different tasks, and you presented, and then you got the fMRI data, right? So you would get this. So then what you have to, the challenge you face is to identify what are the two responses you got, right? So that would be x1 and x2. But the assumption we make is that n is much greater than m, right? The number of independent measures that you are trying to find out is much less compared to the number of observation, right? So again, just as an example in a linear equation, right? So if you have x plus y equal to 2 and x minus y equal to 4, you can solve for x and y, right? So for two unknowns, you need at least two equations, right? And in here, what we are saying is that if you have the number of observation, it is always much higher than the number of unknowns that you seek to find out, right? So if you have two, in this case, a linear transformation can be interpreted as rotation of the data set, right? So you have one data set, these are scatter plot, and then you use a transformation which will make it the slope to be 0, right? So for this one, you would simply use a linear transformation. So principal component analysis, I think as we discussed yesterday, is a linear transformation used to transform data of one set of variable onto another set of variables. And the objective is to produce a new data set that is smaller than the original. Okay. And PCA provides information on the true dimensionality of the data set, and PCA can be used to determine if the data can be recombined into fewer set of variables that still contain most of the essential information, right? So again, in this case, remember I gave this example that, you know, your mom would ask you to study hard, and therefore the amount of time you put into study may be correlated to the grade that you get, how well you do, right? But you can think of that there are many factors, right? Like how much, the amount of time you study is one. Second could be you know, um, um, did you completely cover the course material that was needed, right? And could be, what could be the other factor that would help you get good grades? And it's not that you studied every day, how many hours maybe is another one. So not just you cram right at the end, but if someone was studying throughout the semester, and maybe that would be a better predictor, right? So you have all these measures, but on the other side, right, maybe how you did on your class homework is one predictor. How you did on the, do you have midterms now? Okay. So because like when I was like during my undergraduate, like the class homework was 20%. So the final was 80%. So like you could goof off during the semester, and as long as you studied well at the end, you were okay, right? Um, so so now I guess the weight has become different, right? So before, like all your assignments, midterm, everything accounted for only 20%. The final had a huge amount of weight, right? And if for some reason, let's say you fell sick, then your final grade and the grade would be affected. What I'm trying to say is that these are like different measures, right? And they would carry different weighting. And we have to find out that can we tell what the weighting is and what are these different parameters, right? So PCA basically transforms the data um, into a data set until the variables no longer correlate with themselves, right? So you find a set of orthogonal variables, and then the uncorrelated variables provide no information on one another, but they are not necessarily independent, right? So as I mentioned, like with principal component analysis, each of the variables are orthogonal. However, orthogonality doesn't mean that they are independent. Right? So like here is an example. If you have two time series, right? These are two time series. And this one, would you say that this is correlated, you know, and if you did a scatter plot, the correlation is 0.77, right? Fairly good correlation. Can anyone tell how much of the variance does this account for? Anyone? Hmm? No. No point. So the amount of variance it accounts for is zero point 0.7 squared, like 0.77 squared. So it should be about 50%, right? So this correlation can account for 50% of the variance, okay? So that would explain a lot of the variance, but it doesn't explain all the variance, right? So, but then you can remove this effect and then redo, like, then if you do the correlation between them, the correlation will be 0, right? And the way um, we do the implementation in MATLAB is called SVD. There is a command called PCA also, which will compute the principal component or so this does the singular value decomposition, which will identify the eigenvalues, right? So from the eigenvalues and eigenvectors, you can find how the data would be structured, right? And if you have two data set, then you can generate them. It should be y1 and y2, right? So this is, again, I'm going to give you the slide so you can, if you have MATLAB or any other software, you can run it quite easily. So this is basically a sine wave, and then we are rotating it, and so we would get two time series, one sine wave, the other one which is rotated by 45 degree. And then the question is, if we use this using principal component analysis, what would we get, right? And again, as I said, here the assumption is based on the number of time series that you collect, the number of component is much smaller, right? So you can use this and take the sum of all eigenvector must account to all the variance, right? So this is one way. If you have a several time series, you can find the eigenvalues and eigenvector, and as you add them up, that will tell you how much of the variance is getting added up, right? So if you have a bunch of time series and the first 5 eigenvector account for 90, then it would mean that the first 5 eigenvector account for 90% of the variance of the data, right? And therefore only 5 time series is enough to describe this data. And when you can plot the eigenvalues against the order number, and it's called a scree plot, right? It will tell you where, or it cannot tell you, it will give you an idea where to set the threshold, right? So these are, so this is just as an example. So we are generating 2 time series, right? So this is like this is a sinusoidal, this is a triangular wave, right? And what I have done here is, can anyone tell what I have done here? So I'm generating 1, 2, 3, 4, 5 time series by adding a linear combination of this one and this one with some noise, right? So this one is mostly noise. This one, so you can kind of tell, right? Now that you know how I generated this, you can kind of tell how I, like, maybe this one has more weighting from the sinusoidal, while this one has more weighting from the triangle, right? So this is how I had generated it, right? So in MATLAB, the command to generate is called sawtooth. So the first one is point, right? So this 0.5y plus 0.5x plus a little random noise, right? That's D1, D2 is 0.2y, right, which is sawtooth, plus 0.7 sinusoidal signal and a little bit of noise, right? D4 is more sawtooth, less sinusoidal, but a little more noise. And D5 is mostly noise, right? So this is mostly noise. This is more sawtooth. This one is also more sawtooth. These two have more sinusoidal, or I would say one of them is sinusoidal and sawtooth with equal weightage plus a little bit of noise, okay? So if I gave you this data, would you be able to tell that this data is composed of a linear combination of these two signals, right? How can you tell? right? Can you do frequency spectrum analysis to find out? Or can you use some other method, right? So to do this, we have to use principal component analysis. There are other ways to do, but the easiest way is to do a principal component analysis of this data, right? And so what are the assumptions? You have 5 signals and there are 1,000 time points, right? So the assumption we are making is that the number of sources is much less than 5, right? And also the number of time point is much greater than 5, okay? So this is how you can do like principal component analysis. In MATLAB, those of you who are familiar, there is also a command called PCA, right? So this is called the scree plot, right? And from the scree plot, what you can see is that the first component is this one, the second is this, and then it drops, right? So the first and the second component account for like 67 or so % of the variance, okay? And so the scree plot is that this is where there is a bifurcation, right? And so you take this as that, okay, there are only 2 signals that are present, okay? And also it suggests that the last 3 components account for only 4.8% of the variance, right? And so by doing a principal component analysis, I can identify my X and my Y, right? So I can identify the source. So plot of the first 2 components and the original 2 sources, even the 2 signals are uncorrelated, they are not independent since they are clearly still mixtures of 2 sources, okay? So that is principal component analysis. So it assumes that there are a few dominant signals and every signal that you observe is a mixture of them, and therefore by using principal component analysis you can use to find the number of sources and then decompose to find these signals, right? So based on what we have learned so far, if we were to give fMRI data, right, the time series data, what would we get? Let's first think of the finger tapping experiment, right? So for the finger tapping experiment, if I took the whole brain and fed it to principal component analysis, what would be the main sources? Any guesses? So what I was saying is that if you took the finger tapping data, right, where the subject tapped the finger for some time and then left foot, right foot, tongue, right, that data, if you fed to principal component analysis, what would you get? So maybe, okay, so let me try to make it maybe a little simpler first. So there is activation in the sensory motor cortex and there is time series. What would happen if I took a region from the sensory motor cortex and then looked at the time series. How would they look like? So, anyone? Maybe should I? So, okay, maybe I'm not saying it properly. So let's say um, you scan two subjects, right? And did they tap their fingers? Okay, so let's say they had tapped their fingers for like 20 seconds off, 20 seconds on, right? 3 times. And after that, you um, like use a software to look at the data and then wanted to look at the sensory motor cortex and you clicked on that region. How would the signal look like? Mm, okay, let me do this. I want all of you to understand, so let me just okay, so this is when the person, this is a person with brain tumor. You can see the tumor in this region, and this is, they tap their fingers, right? They tap their fingers for 20. So this one, they tapped their fingers for 20 seconds, then they stopped, and then they tapped again, then stopped, and then they tapped again and stopped, right? So if I took, right, so this is like a 3 by 3 in the motor location, right? So this is like, if I displayed like the way I used, so this is one time series, this is another time series, this is third, 4, 5, 6, 7, 8, 9, right? So you have 9 time series. Now if I did, this part is clear to everyone, right? So you have 9 time series. Now if I gave you this 9 time series and asked you, tell me how many sources are there, right? What would be your guess? Is it 1, 2, 3? Joshua? No, no, this is finger tapping. This is finger tapping. Hmm? As I said, because the response could be due to underlying hemodynamics. It could be due to, right? So different voxels are weighted differently, right? It could also be that that is not from the motor region. It is possible that this voxel is not in the sensory motor cortex, and therefore there should not be any response, right? Is that clear? Okay, so now you have 9 time series. Now tell me, if I ran a principal component analysis, what would I get? No, this is 9 time series. Okay, good. So if I ran principal component analysis, I would get nine like eigenvalues, right? But how many of them would be significant? How many sources do you think is present in this data? How much? You think there are 3 sources here? Okay, so that is not the question I'm asking. Okay, maybe I'm not saying it correctly. Okay, so so you are given, I give you this. Uh, okay, never mind. Okay, so let's say, okay, so this is 6. I'm giving you 9 time series, right? You have to tell, on this data, we got how many sources? 2, right? The data I showed you, how many sources are there? Say that. Oh, you want to see the data? Okay. One second. Control. Okay. Look at the data again and tell me how many sources are there. Oh, never mind. Red one is, I was playing with the data. Very okay. You would get 9, right? But how many sources are there? 1, right? Because that one is like this one, right? Because this is the sensory motor cortex, and that was the task that was given, right? They did during this, and that's why, depending on the location, there is a different linear mixture. Is that clear? Yeah. This is clear to everyone, right? So now is if instead of taking this 9 voxels, if I had used shift if I used this as, so now you have maybe 50 time series, right? And then you were given this, how many sources are in this data? Uh, maybe, but okay. If you look at this, do you see another pattern, right? So what happened? 4. Okay, I don't know what it is. So this is what I'm trying to say is that depending on your location, the number of sources can vary, right? And this one is like for the motor, because it was motor, it was like good. But if it is, um, you know, let's say if you click on the tumor region, right, the response is going to be quite different, right? So if you take the whole brain wall, if you know exactly where it is, then it is easy to identify, like it could be 1 or 2. However, as you take the whole brain volume, then this one may not be the most dominant one, right? Because maybe the respiration signal, maybe the head motion, maybe some other factor may become the most dominant one, right? And what I'm trying to say is that this is where we can use principal component analysis to find out how many dominant sources are there, right? And based on that, we can identify, what is happening. Okay. Okay, let's go back to, right? So again, from here, we can find the original component, and then you can basically, because that was a 5 by 5, like there were 5 time series, you can generate a covariance matrix which shows the relationship between them, right? Again, suggesting that the first component was the most dominant component, then is the second, third, fourth, and fifth, right? Mostly noise sources, um, is present. So this is, again, like um, the main difference between principal component and ICA is that in principal component analysis, the different sources it identifies are orthogonal to each other. However, orthogonality doesn't mean independent. It means that the correlation between 2 time series is 0. However, they could still be independent. They could still be dependent, right? So ICA is a new way of analyzing data, or new method. In this, the idea is orthogonality is not enough. We want to make sure that the different components are independent of each other, right? And this is one of the first studies that was done in the 1980s using neurophysiological data, where they had used, right? And as I had said, like the comparison that ICA is typically used is called this cocktail party problem. So again, I think it's like saying that imagine you are at a party. For you, it is no problem to follow the discussion of your neighbors, even if there are lots of other sound sources in the room. And the discussion in English and in other languages, different kinds of music. You might even hear a siren from the passing by police car. And if you think about it, um, you like the brain doesn't have a unique filter based on time or frequency, so it uses different processing strategy to extract this information, right? And it is not known exactly how humans are able to separate the different sound sources. Independent component analysis is able to do it if there are at least as many microphones or ears in the room as there are different simultaneous sources. So again, there is this website from Finland where one of the most commonly used toolbox called Fast ICA was developed. They give some very good examples, right? So ICA is a method for finding underlying factors or components from multivariate data. And what distinguishes ICA from other method is that it looks for components that are both statistically independent and non-Gaussian, right? So they are independent, non-Gaussian. And so you can, like before, you can write it as, you know, if X is given by this, and this is the signal, and this is the unknown matrix, right, that we have to find. How are these? We don't know what the input is. We only know the output. We also don't know what the mixing matrix is, right? The mixing matrix is, right, this is the mixing matrix, right? We don't know that, so we have to find out how these signals were generated. Okay, so we have only this, right? So we have to find the mixing matrix and the sources, right? And for linear noisy ICA, when assuming uncorrelated, then this can be represented as X equal to AS plus N, right? And this independent component analysis is related to principal component analysis, factor analysis, also a blind source separation method, right? Because we don't know the mixing matrix, we don't know the sources, we only know the observation. So this class of analysis method in signal processing is called blind source separation method, and it has many applications in the last 20 years or so, right? So independent component analysis seeks to transform the original data into a set of independent variables. The motivation is primarily to uncover more meaningful variables and not to reduce the dimension of the data set. When the data set reduction is also desired, it is usually accomplished by pre-processing the data set using PCA. So like in ICA, we don't know how many sources there are. So the way we find the sources is by initially using the principal component to find what the number of sources and feeding that to the independent component analysis to find the sources. Again, as this is just a simple representation, right? So you have mic 1, mic 2, and then number of speakers. And then by listening to this, we should be able to or we have to find out what it is. Right? And again, this is the representation where from the observation we have to find the unmixing matrix and the sources. Right? And so some of the limitation is that ICA can only be applied to non-Gaussian signal because it relies on higher order statistics. The higher order statistics, that is moment and related measures of Gaussian signals, are zero. Also, since ICA has available only the measured variables, it has no information on either the mixing matrix or the underlying source variable. So there are some limits to what ICA can do, right? So this has to be non-Gaussian signal distribution. So ICA cannot determine the variances, hence the energy or amplitude of the actual sources. So in principal component analysis, you can tell if there are two sources, what was the magnitude of, let's say, triangle signal versus the sinusoidal signal. In ICA, you cannot. It will only tell you the signal, the magnitude is missing. Okay? These are some of the pre-whitening methods that has to be done. And there are several ICA algorithms that have been proposed for analyzing the data. They basically use an objective function which is related to the independence of the data. Also, the various approaches that is used differ in the specific objective function that is optimized and the optimization method that is used. And one of the most intuitive approaches uses an objective function that is related to the non-Gaussianity of the data set, right? So these algorithms assume that the data is non-Gaussian. And this approach takes advantage of the fact that mixtures tend to be more Gaussian than the distribution of the independent sources. And mixture of non-Gaussian sources will be more Gaussian than the mixed sources known as, like in the next slide, right? So like these are, I think we had covered, this is the Gaussian distribution, but there are other kinds of distribution like single sinusoid distribution or two sinusoid distribution, quadratic sinusoid distribution, and so on, right? So again, as long as our data is generated from these kind of distribution, we can use ICA algorithm where they focus on non-Gaussian properties such as kurtosis or the cumulant of a variable, which is zero in a Gaussian distribution, to estimate these, right? Again, there are many different approaches to perform ICA algorithms. Like in MATLAB, there is JADE. Also, there is something called fast ICA, which is also very good. You can try either of them, right? Right. So in this case, again, the same very similar example, right? We have a sinusoidal signal and the triangular signal, and then we just add, like, we randomly add them up, right? And like these are the different like original component before mixing, and then we use IC, we then add them up like before. You have 5 different signal that you get by adding the signal sources that I showed you. Then you use the scree plot, and in this you can see that the bifurcation occurs at 3, and then by using ICA, you can identify these 3 signal sources, right? Quite amazing, right? You have no idea what the signals are. You randomly add them up, and then by using ICA, you are able to identify, right? So that's the basic concepts for ICA. And so you can imagine it has a lot of application. The different applications are not only for identifying the signal sources, but also to clean up your data, right? So, if so if you did this and you had like 50 or 60 and the scree plot was 3, then it suggests that the other sources are mostly random noise and you can exclude them, right? So it is widely used in EEG, like most neuroimaging modalities I know, they have used this. Okay. Any questions so far? No question? Yes? Right. Yeah. Right. So you use principal components, right? So you say that, okay, if principal component is saying there are 10 sources, then let's start with 10. So that is one way, but there has been other approaches where people change the number of like, so let's say principal component says there are 10 components. People have also tried, they will try 10 first, then they may try 5, then 12, 15, 20, and then they will run it many times, right? So then they will see with 10, like 15 and 20, are there any components which are common, and which are the most common component, right? So you can like run this like thousands of times to find the sources. Okay. Any other question? No? Okay. All right. So okay, so it's 11, so let's talk. Okay, so ICA was first introduced by a very famous neuroscientist called Terry Sanowski in 1998, right? And so they used this on fMRI data to show that you could separate these components. And so they had a paper, right? And just during that time, I had also published a paper on ICA using task activation. And this is what I had done, right? So I had made the subjects tap their finger, and in between, we would give them carbon dioxide, right? So the signal went up, right? The signal went up. So when we did principal component analysis, it could not separate them, but when I did ICA, we could separate this, right? And I always wanted to use this for resting state data, but I could not find a way to use it, right? And the reason was, in the MRI data, the number of voxels is large, right? But the number of time points is small. And according to principal components, the number of observation should be much larger than the number of components, right? So I tried, and I could not find a way till a paper was published by Vince Calhoun, right? And this was in 2001. And what he showed was that if you have the data, this is the number of voxels, and this is the number of time points. So this is your data matrix. And then you can use ICA to determine your mixing matrix, and then you can find the spatially independent time courses, okay? The other way he also did is that this is time, this is voxel, so you can do principal component analysis. The other way he did was, this is time and this is voxels, right? And then he used this to derive this temporal component, and basically to show that you could do ICA on the spatial map, and what you get is exactly the same as this one, right? And therefore, doing ICA in space is the equivalent as doing ICA in time, right? Because in linear algebra, if you're trying to find the eigenvalues, if you have a matrix, the eigenvalues will remain the same whether you transpose the matrix or not, okay? And this was a very important observation. And by doing this, so far we have said that any time series is a linear combination of different sources, right? But what he's saying is that any brain image, right, is a linear combination of a set of brain states, right? And by formulating the problem in a different way, he could use time as the observation and the voxel as different measures. And from that, they could extract these things, right? So this was the two tasks that was presented. And right, so these are the two tasks that was presented. And from these tasks, right, these are temporally and spatially uncorrelated. So there is no dependence between them. These are like temporally dependent, right? These are spatially dependent based on the stimulus, and these are both temporally and spatially dependent, right? So that is how they had designed their task, to show that they could use ICA to identify these things, okay? And what they could do was, right, so with spatial ICA, they could find the components, this is one component, this is the other component, and that they were spatially dependent, they were temporally dependent in cases, and then they were spatially and temporally dependent, right? And using temporal, they could also show this exactly the same thing, right? And this paper basically showed whether you do the data analysis in space or in time, you would still get the same result, right? Again, so this is spatial ICA, right? And if you did regression, you would get this. If you did temporal ICA, you would get very similar result. So these are temporally and spatially uncorrelated. This is spatially dependent, right? Again, this is using GLM. This is using temporal ICA. And again suggesting that the results are very similar. Then they wrote, maybe, okay, before this, let me ask, is everyone clear? Any question? So for resting state, so all of you remember about fixed effects analysis versus, right? For resting state data, can we use fixed effect analysis? So if we have 10 subjects, can we do fixed effect analysis? Or do we have to use random effect analysis? Can you do fixed effect? What does fixed effect mean? You take the fMRI data and average across 10 different subjects, right? Now, if there was if they were all doing the same task, when you add them, that task becomes prominent, right? But with resting state, can you do fixed effects analysis? What would happen if you added them up? So let's say all of, so you have finger tapping, let's say finger tapping paradigm. All of you performed the same way. If we added all your brain into an averaged brain, then if we looked at the sensory motor region, right, we would see like a very nice motor activity, right? But can you add different, what would happen if you add, let's say we scanned your brain 818 times, everyone's brain 818 times, and then you added them up, what would you get? What would happen? Okay, so if you added task, the noise would cancel out and only the task would remain, because that is common to every subject. With resting state, are you giving any task? So then what would happen if you averaged subjects? So you would have like, yeah, you are just adding noise to noise, right? Like your resting state signal and let's say someone else's signal, there is nothing like nothing in common. And let's say, um, just as an example, let's say even if your heart rate was the same, like two people's heart rate was similar, it's very unlikely that it will also be in phase with each other, right? When you collect the data. So it would just cancel out, right? So we cannot use like the fixed effect analysis, right? Hmm? Right, right, right. So it like it would just be noisy and whatever correlation may be there, it would be destroyed, right? So the fixed effect analysis would not work for resting state data, right? So um, this is showing um, maybe, no, that's not what I said. What I said is the time series is going to be very different, right? So you cannot add them up. Mm, I think um, I want to convey this information because it's very important, but I'm not able to convey. Mm, maybe someone can say, yeah. Right. Um, yeah, they are noise, but they noise in a sense that so okay. Let me. I'm not explaining. So let's say five of you. I collected MRI while five of you tapped your finger, right? 20 seconds on, 20 seconds off. Now I could take a time series from your brain, from the motor cortex, and then use that as my seed region, right? And use it on, on the other four. And I would still be able to activate the sensory motor cortex, right? However, if I took your resting state time series data from the sensory motor cortex and tried on the other four, there should be no correlation because because uh, di different what right. So all the source, there is nothing common, right? The heart rate, respiration, and even if the heart rate or respiration is similar, very likely they will not be in phase with each other, right? In right. So do you? So maybe the other question I would ask is that let's say um, we like who was scanned that day. So, so let's say Rahul was scanned that day, and we scanned Rahul again today after lunch. Can we use? Can we average his data? Can we? Can we not? Hmm? No, we cannot, right? Because these are temporal signals, and they will have, again, even if the heart rate and respiration is similar, well, first of all, it's very unlikely that they will be similar. There will be some changes. They will be out of phase, right? They may not be, it would be very unlikely that it would be in phase. So it would be like adding two random signals, right? So you cannot average the data in resting state, right? Yeah, no. So in that case, all the data was processed and concatenated and then analyzed. Because we cannot, because if we average, then we are throwing away the, like, yeah, yeah, right, right. So it's hard to say which one is better, but what you can say is that maybe for task based, if you know, for task based, you have some idea as to how many sources there are, and therefore maybe principal component is adequate enough, right? Like for finger tapping, I don't think there is going to be any difference between ICA and PCA, but um, for other complex tasks, right? Let's say um, like five of you watched um, uh, like, which one is your favorite movie? Hmm? Which one? Okay, Rahul. Huh? Okay, besides that, I'm asking because I want to, I want you to tell a movie. It would be like only like young males would like, and no one else. And huh? Animal. Which one? Like animal. Okay. Okay. And which one would you like? Which is your favorite movie? Okay. Okay. All right. Okay. So then let's say we made them. I thought, okay, I like Three Idiots also, right? But so uh, think of another one which maybe Rahul would hate. Anyone? Okay, Dracula. Okay, is it a new version? Okay, okay. All right. So anyway, you get the idea, right? If um, we made you watch two different clips, right? One which, if there is such a thing, only males would like, versus another only females would like. But we made like five males and five, our population was 10 subject, five male and five females, and they both watched 10 minutes of a male, like male centric movie, versus and 10 minutes of a female centric movie. What do you think would happen to the activation? Would, can we use, can we average the signals? right? So in that case, maybe principal component analysis may not work that well, right? Um, and in that case, it would be good to have all these runs and then use the ICA or a PCA to find if there are specific features in the specific brain regions which only she would like to specific movie versus in the other movie which maybe Shreyans would like, right? But in those cases, there is still a common component, right? Which is like the visual or auditory, um, which would be activated in all the subjects, right? So that is how, like, whether you use PCA or ICA would be relevant, and perhaps like ICA. But again, so in resting state, because these are, again, I should say random signal, right? These are, I should say, spontaneous signal unique to a person and the time when the data was acquired. We cannot average it, right? Because there is, when you average two signal, you assume that there is a signal and a noise component, excepting in resting state, there is, the signal component is not known, right? And because it's not known, if you add another signal, you're basically canceling it out, right? So therefore, you cannot use the analysis like we do for task activation. So what is done is like shown something like here. You have subject 1, like subject 2 and subject like M, and what we are doing is we are using these measures to find the components corresponding to each subject, right? So like the sensory motor cortex that I showed you before from the first subject, then you process for every subject and then try to aggregate them, right? That is one way. The other way is that you have these components corresponding to each subject, and then you do a group average, right? So that will give us the components. Is that clear? And so they have shown here that in one condition, there are two spatial map. In the other condition, there are three, okay? And right, so just like before, they have flashing checkerboard, right? This is the right side, and this is the left side. This is presented, and then by doing this analysis, you can basically make group level activity map, right? Using ICA for task activation. And again, this is showing the similarity between GLM analysis and ICA back reconstruction. And then from back reconstruction, you can go and look at each individual ICA map also, right? And then you can find the time series corresponding to this, right? So this is from 9 subjects, and then you can find the temporal components for any given subject, and this is how it looks like, right? Like these are the two paradigms, the third, fourth, like fifth, most likely are noise sources. It could be related to head motion or respiration and so on, right? So this was a paper by Vince Calhoun. This is the paper by Christian Beckman. And what they did was, so you have a resting state data, and people would like take data 1, data 2, data 3 and do ICA. But what they did was they used concatenation, right? So data 1, data 2, data 3, so on. Got it? So the number of time points in fMRI is small, but the number of voxels in fMRI is large, right? That's why we had to struggle whether to do it in space or time. So to overcome that problem, what they did was, let's say each subject had 200 time points, and they found like they got 100 subject, so 100 times 200, right? So 200,000, while the number of voxels is maybe 10,000. So now you can do a component analysis along the time domain, right? And then by doing that, they could identify these regions, right, from one resting state scan. Okay, and it's of course very hard to prove. I think, as someone had said, that each one, how do we know each one corresponds to what, right? So like, but some of these networks look familiar in a sense that this one looks from the visual, this is auditory, this one is sensory motor, right? Visuospatial, dorsal, visual, right? And so on. So by doing this, one could identify, again from a single run, different resting state parameter. Again, this is another paper where they also were able to identify a number of components, again, like visual, where is the sensory motor? Can anyone tell? Right. Right, this one, or this one. Like H looks like that. So by using this, one can identify these networks. And again, with resting state, we are taking the time series, decomposing them into the spatial components, and then identifying them, right? So when it is done in time, it is called temporal ICA. When it's done in space, it's called spatial ICA. But as I mentioned, that they are quite interchangeable because the eigenvalue of a matrix doesn't change if you transform it, right? And whether you are looking at the data, right, across space or across time does not matter, right? Any questions? No? Okay, so in that case, so what would you get if I asked you to do PCA on a resting state data? Anyone? So what would you get? What? No, that I know, but where, like, if you, but what would be those sources? So, okay, so, okay, we have a little time, so okay. So this one, all of you agree, right? If I took the 9 series, you would get one source, right? What would happen if I took all the voxels and did principal component? What They would be independent sources, but what would be the sources from? Okay, well, the tumor region looks like this, right? Um, so maybe the tumor region has a signature, maybe, right? We don't really know. We can test it. So let's say what you're saying is true. So one would be from tumor region, the other would be from the motor region. Any other sources? Uh, the visual, the resting state visual, the visual component. Okay, so the visual is here, right? So it would be like this, right? Hmm. Okay, so those are also okay. Heart rate, maybe something, right? Something like this, or even like this, right? So uh, if we took the whole brain, what are the main sources of noise that we had discussed? The the most source of the most common source of noise is head motion, right? So most likely the first component would be head motion, right? And the second or third would be physiological noise. Very good. So it would be like respiration signal. It won't be heart rate because the heart rate is aliased. We cannot see the heart rate, right? So generally the first few are like head motion, respiration, other noise sources. Then would be signals of interest, even for task activation, right? Because the motor has nice signal, but it won't be the first or the second because uh, right, good, but the motor region has good signal response, right? Like the motor region had good signal response, so why can't we still, right? So this has good response, but why can't we see good, why wouldn't it be one of the main components? Right. So this one I haven't done any correction, right? So the motor, it's unlikely that it will be one of the first few components because look at the volume of the motor region, right? So the motor region is a small fraction compared to the whole brain. So that's why it's unlikely that it will be the first component, right? So another limitation of ICA or PCA is that, like in GLM, you know what the signal source is, so you model it and you see the response, right? In this case, you have to look for these components, and by looking at these components, then you have to decide whether these are signal sources or noise sources, and then decide. Is that clear? Is that clear? No. Okay, so what I'm trying to say is that in this case, right, in this case, the voxels in red are the voxels which are active, right? The other regions, they are noise, right? And our task was for them to tap their fingers, right? But the sensory motor cortex, although the response is good, maybe is, would you say it's 5% of the whole brain? Maybe, right? Maybe, may not be, right? So even if 5% has good activation, for the rest 95%, if the respiration signal is there and contributing, that would be the main component, right? And not this one, right? Although this is our primary signal of interest, right? With GLM, this would be our primary source of signal, and because we know that these subjects, this is what was given to the subject, this would show up. Is that clear? Let me just try something. 3D. Okay, so maybe in the afternoon when we perform this, the data, I can show you how with ICA, the results that we get with resting state, and then what would we get with PCA, right? But just remember that with resting state, like each signal is different from each subject. Each signal is different, although not all the signal in the time series is noise. There are noise components which are present, and that is where the challenge of resting state is that you don't know. There are these spontaneous activity which is present, but we don't know which one, like what fraction is noise, what is signal, and therefore we have to basically concatenate them for group analysis and perform ICA, or for single subject, we also use ICA to find the different components, and then we can find the spatial component, and from the spatial component we can find the temporal component, and from temporal component we can find the spatial component. Okay. Any questions? I think I just confused you all a lot. But maybe once we look at some data, you will have a better idea, I hope. Rahul, you had a question? Okay. No question? Anyone? No question? You're ready to analyze the data? So, so there are different analysis method, right? So far we know about seed-based analysis. What is seed-based analysis? So you take a time series from one region and correlate with the rest of the brain. So that region that you take the time series from is the seed, and then you find out which other region has been activated, right? So seed-based region analysis is called like hypothesis-driven analysis, because you have some idea. Like in this case, let's say in this case, my hypothesis is that the sensory motor cortex, the left and right, are correlated, right? Right, so here, so if I, right, so if I, like as I move my pointer, the region which shows up are correlated, right? So these are the regions which are correlated, and this one would, so this would be seed-based analysis. So this is hypothesis driven, right? I have some hypothesis, and for that hypothesis we can test, right? What kind of analysis do you think PCA is? Hypothesis based analysis? Shivani, is PCA hypothesis? Yeah. Okay, how about Arundhati? Very good. So PCA and ICA are data driven. So what is the difference between data driven and hypothesis driven? From the data. Very good. So with hypothesis driven, you have to have some idea as to which region is connected to which region or how the sequence of event occur. While in data driven, the results hopefully will inform you about how the brain is structured. Right? Any other questions? Yeah, it is. So it is data driven, right? So it's an exploratory analysis. Well, without maybe saying without requiring any knowledge is too strong a statement to make, but maybe without having a very specific hypothesis, you can analyze the data, right? So with ICA or PCA, you don't even have to know how many signal sources are there, right? And then you can analyze and then find that there are two signal sources. In hypothesis driven, you have to, like, you must know that there are two, right? And so sometimes people use hypothesis driven, but sometimes people use data driven, and then based on the results develop hypothesis and then do the analysis. Any other question? Okay. Sorry, we stop now. Okay. I'll try to answer any other question if anyone has, but okay. Right. Yeah, yeah. So you can, so for the task-based, you can present the data in a different way, right? So right, so let's say you have like voxel number 1 till voxel number 50, and they all behave in this way. So maybe we can end here. So we have 50 of this.